So let's go ahead and get started. And it's a privilege here today to have Patricia Schulte with us. She's a professor at the University of British Columbia, the Department of Zoology. She's had a very distinguished and prolific career, publishing more than 200 papers and counting, and is an elected fellow of the Royal Society of Canada, which is the equivalent of the National Academy of Sciences here in the US, and received the Award for Excellence in the Society. She started her career back where she is right now. She did her bachelor's and master's there before venturing south to Stanford, where she did her PhD with Dennis Powers. And her work is characterized by integrating physiology, genetics, and genomics to try to understand how organisms respond to and adapt to variation in their environment. And I met Trish last year she was a keynote speaker at a Gordon conference on marine molecular ecology, where I first got introduced to this idea of epigenetics and how this may allow things to adapt much faster to the environment than the types of genetic adaptation that I learned when I was a graduate student. So without further ado, thank you for being here.
Well, thank you so much, Paul, for that extremely kind introduction. And thank you all for coming and attending this talk. It's a real pleasure to be here. And I'm just sorry that this has to be a fairly brief visit because this is a teaching point for me, so I have to run back and teach a class on Friday. Ordinarily, I try to combine multiple trips to reduce the carbon footprint of flights. So for this trip, I couldn't do that, but I did purchase carbon offsets, so that at least in part mitigates. Because I think that's one thing as academics we really need to think about is the environmental impact of our activities. And our field work sometimes takes us to far-flung places, but we should always consider carefully what we're doing and the impact we're having on the environment as we do it. And the reason why I say that is that the research in my lab really revolves around understanding how animals cope with environmental change. And I work primarily on fish. And this kind of encapsulates the way I think about these problems. So I'm really interested in understanding how variation at the level of the genome integrates up through various levels of biological organization to influence the performance of an organism in a changing environment. And so today I'm not going to go dig too deep into this mechanistic detail here in the middle. We'll mostly be leaping from here up to here, but I just wanted to make you aware that a lot of the time and effort in my lab is actually spent in digging into that kind of mechanistic detail, but always in the context of understanding the implications that it has for how animals perform in their environment. And for many years, I mostly thought about this in the context of responses to natural environmental variation. So either spatial variation, so here you're looking at temperature variation across the globe. One of the things we're really interested in understanding is what are the abiotic factors that help to set species distributions and abundances across the landscape. Of course, abiotic factors are only one of the many factors that can influence where animals live. And then also too, we've had a major focus on looking at shorter term temporal variation, so seasonal variation or variation with the tidal cycle, and understanding how organisms cope with that kind of change. But with this kind of perspective, it's hard to escape thinking about anthropogenic change, because we know that environments are getting warmer. So not only are there increases in average temperature, but there's also more extreme heat, more extreme hot weather. And for aquatic organisms, that increase in temperature is accompanied by decreases in oxygen and also acidification. And all of these things are presenting us with a problem. It was presenting animals with a huge problem, and us with a problem in terms of how are we supposed to predict and manage these changes. Of course, we want to mitigate the environmental change as much as we can, but we are on track for a changed world, even if we actually manage to get our carbon production under control. So
when we think about how animals respond to anthropogenic environmental change, there's almost kind of a meme about it. The idea is that populations can either migrate, so move away from areas that are no longer suitable, so that in fact it means a local extinction, or adapt and die. This is sort of like, if you go online, this is sort of the meme that comes up. In fact, to the point where you can go and buy yourself a mug with that on it. And the irony of selling stuff to talk about a problem that is ultimately caused by our addiction to stuff doesn't escape me, but this is something that we need to bear in mind. But as biologists, we also know that this is way oversimplified. So this idea of migrate, adapt, or die, that adapt word has a lot of subtlety inside of it. And at one level, we can think about that adaptation, as a biologist, when we say adapt, what we really mean is evolutionary adaptation. But physiologists and ordinary people often talk about adaptation within the lifetime of an organism. And that really represents phenotypic plasticity or epigenetics. And then epigenetics also shades into things that go across generations. So we can think about this as plasticity, epigenetic variation, and adaptation along a kind of a temporal scale, where plasticity occurs on scales of seconds to minutes to seasons. And then development would be across the lifespan of an organism, so that's beginning to be an epigenetic sort of change. And then inheritance from one generation to the next. And then ultimately to genetic change in populations, which inevitably is at least one generation, and generally speaking, much more. In fact, when I was a grad student, we pretty much thought about genetic change in populations and evolutionary adaptation as something that takes place relatively slowly. In fact, now we know that adaptation can be quite rapid. But fundamentally, this is sort of the mental schema in which we work, because these at the very fastest responses, they would be behavioral. So an organism can move if it's the type of organism that can move. Then there's physiological changes during development, changes across inheritance, and then changes in populations. And what we're going to do through this talk is think a little bit about each of these things. And I'm going to warn you right up top that this is not going to be the most satisfying talk ever, because I'm going to draw from various parts of work that we've done, but it was never conceived in this as an answer to this full schema. So there are many places where we'll just kind of say, that's as far as we got, we don't have an answer for you. But I think this is useful as a way of thinking and as a way of shaping our thinking. So if we want to think about how organisms cope with environmental change, we have to think a little bit about what we might want to measure. So how would we assess whether an organism is likely to be resilient or likely to have problems? And there's a variety of ways you can do this, but one way that's quite common in the thermal biology literature is to think about a thermal performance curve. So a thermal performance curve is that thing that's shown in the middle panel there, where on the x-axis is the body temperature or the environmental temperature of the organism, and on the y-axis is some performance trait. So that could be anything from the rate of a biochemical reaction all the way up to the intrinsic rate of population growth of a population. So you can measure performance at many levels. But generally, whatever level you look at, the relationship between temperature and performance is some kind of hump-shaped curve, often with a skewed distribution with a sort of a shallow increase as temperature increases, and then once you hit a maximum temperature, tends to have a relatively steep drop-off. And the position and shape of TPCs vary among organisms. So the bottom part of the panel there just shows different kinds of organisms, whether they might be generalists, which have a wide a thermal performance curve, a specialist that has a narrow one, or a warm adapted or a cold adapted form where the curve will be shifted along the axis. And looking at these allows you to kind of make an estimate of whether an animal is likely to be resilient to increased mean or maximum temperature. And one thing I want to point out, because we'll be using it in the talk today, is what's called the CT max, which is sort of the maximum tolerated temperature. And usually the way this is measured in practice in organisms is you take your animal, whatever it is. For my case here, we'll imagine a fish. So imagine a fish in a beaker. You gradually heat the water up in that beaker, you watch your fish, and when the fish turns over, you say, aha, that cannot be good. That's the CT max. Usually this is a non-lethal endpoint. If you take the fish out and put it into cooler water again, it will be fine. It turns out that CT max is strongly associated with the latitudinal distribution of organisms. So there have been a number of macroecological and macrophysiological studies that have showed this pattern. But you might think that it's kind of a weird metric, you know, how often do animals just kind of get boiled to death? And the answer is more than you would think. This photograph here is a bunch of dead bats in Australia. So this was a heat wave, and it was hot enough that the bats just fell out of their roosts and ended up on the ground. Generally speaking, they could be rescued, and if they were cooled down, they would be alive. Here's a photo of that green stuff down there. Those are dead, or almost dead budgies. Again, heat wave in Australia. They just fell out of the bushes. Now, if we go back a slide, you can see that there's a CT max, there's also a CT min. And remember that as climate changes, we're actually likely to get more extreme temperatures. So low minimum temperatures may also occur. And I just pulled down a picture from the National Weather Service. And this is actually from yesterday. So this is falling iguanas possible tonight. This is in Florida. And you can see in there, it says iguanas are cold-blooded. Cold-blooded, of course, is a misnomer. Ectothermic animals have blood, or have a body temperature that is the same as an environmental temperature. So when it gets cold, their body temperature drops down to the environmental temperature. And when it gets too cold, they become immobile. So just like a fish turning upside down in a beaker of water, iguanas can no longer maintain the muscle contraction that they need to hang onto trees, and they drop out of the tree and lie on the ground. And you will see that at the bottom it says, they may fall from trees, but they are not dead. And that is true, they are not dead yet. But if we think about this from an ecological perspective, a little immobilized iguana on the ground is likely to be someone's dinner. So people often call CT max the temperature of ecological death. And so although it's an extreme, this would be something this isn't going to happen in response to a change in mean temperature. These kinds of measures are actually relevant in the context of extreme temperatures that animals may encounter in their environments. So what I'm going to do today is I'm going to give you some examples from 3 species of fish that we work on to look at the potential roles of plasticity, epigenetics, and evolutionary adaptation in allowing these animals to cope with temperatures that are different from what they have historically adapted to. So let's begin with the shortest term thing, which is plasticity. And I'm going to start with examples from the fish on the top there, which is the Atlantic killifish, and the fish on the bottom left, which is rainbow trout. So here's an example of plasticity and thermal tolerance for the Atlantic killifish. So what you're looking at here on the x-axis is the acclimation temperature. So this is the fish have been taken into the lab and they've been exposed to a particular environmental temperature for a period of a couple of months. And then we measure the CT max, so we put them in their beaker, we heat the water up, and then we ask what temperature do they turn over at. And so what you can see is, first, these fish are very eurythermal. You can acclimate them to temperatures as low as 2 degrees and as high as about 35 degrees. And then you can see that their CT max increases as you acclimate them. The 2 different lines are actually 2 different populations, and that will come back later. But for now, just concentrate on the fact that CT max is plastic. It changes with acclimation, and so does CT min. So a summer acclimated animal, an animal that is being exposed to warm temperatures in the summer, will have a higher thermal tolerance and a worse cold tolerance than that same animal if you measured it in the winter. So that's cool, but this plasticity has limits. The reason this graph doesn't go any higher than 35 degrees Celsius is not because we got tired and didn't feel like measuring it. It's because if you try to take that fish much higher than 35 degrees, they start dying. And they start dying actually quite quickly. If you hold them at temperatures above 35 degrees for more than a few weeks, you will lose about 50% of the animals. So this plasticity clearly has limits. They can improve their tolerance to a certain extent, but not beyond. And so the plasticity has limits. And in fact, that 35 degrees is pretty close to the maximum summer temperatures that these fish experience at the southern end of their species range. Okay, so there's lots of plasticity, but it has limits. But maybe this is just something about this particular fish, which is adapted to a very variable environment and that has a very broad tolerance range. What about a fish that is a bit more constrained, that's more of a cold water specialized fish? Bottom line is we see a pretty similar pattern even in them. So this is an example from rainbow trout, which is primarily a cold water species. And so here we've acclimated fish to 12, 18, and 24. And again, you can see that their thermal tolerance, again measured as CT max, increases with temperature. But notice that 12, 18, and 24 are equally spaced. They're equal distances of temperature apart from each other. But look at the change in CT max. It's not nearly as big between 18 and 24 as it is between 12 and 18. So this suggests that these fish might be reaching a limit. And so in this species, we've done quite a bit of work to try and understand what the underlying physiological factors might be. And so this is the one place where I'm going to do a little, delve down a little into that black box of mechanism, because I think this data is so cool. So this data will take a little bit of explaining. So in this case, on the x-axis, what we have is exposure temperature. And on the y-axis, what we have is heart rate. So you can measure the heart rate of a fish the same way that you can measure it in a human. Many of you may be wearing a Fitbit or an Apple Watch. Those things are detecting your heart rate just with a surface-mounted sensor, right? And so same thing on a fish. We can put a sensor on the outside of the fish's surface, and we can use that to measure its heart rate. So we take these fish, we instrument them up with a heart rate detector, and then we increase the temperature and measure the heart rate. And so what you can see is that overall, heart rate increases with temperature exactly as we would expect. But you can also see that at some point, heart rate levels off. And then you'll see there's some points here that are not connected with lines. That's because at this temperature, some of the individuals are starting to have cardiac arrhythmias. So they're starting to have little mini heart attacks. These three different lines are fish acclimated at three different temperatures. So this is a 12-degree acclimated fish here in the light gray. And you can see that it's tapping out somewhere around 25 degrees Celsius. Here's an 18-degree Celsius acclimated fish. It's tapping out a little bit warmer. But the really interesting one is the 24-degree fish. This fish, yeah, it's tapping out higher, so it's doing a little better. But notice that its overall heart rate has been suppressed. So this fish is making a trade-off. So in order to allow its heart rate to be capable of operating at higher temperatures, it's suppressed overall heart rate. And in fact, these fish don't feed, they don't grow, and they don't last for all that long at 24 degrees. You might get them to go for 3 weeks or 4 weeks, but that is really the highest temperature that they can go at. And we've used RNA sequencing to look at the cardiac transcriptome. And this is just a heat map. It's mostly here just to remind me to tell you about this. What we see in these fish is that from 12 to 18 degrees, the kinds of changes in gene expression that we see are consistent with beneficial acclimations, so changes that you might expect to improve cardiac performance at high temperature. At 24 degrees, we see mostly things that are consistent with the cellular stress response. So these fish are really struggling at these higher temperatures. So the two kind of takeaways from this is, again, that there is ample plasticity, but it has limits. And once you reach that limit, you're kind of stuck. So in both of these species, with very different life histories, very different kinds of thermal performance, we see there's plasticity in the upper temperatures they can handle, but they're pretty much correlated with the maximum temperatures that you currently find these fish. So rainbow trout, for example, their species, natural species distribution extends down into Southern California. And in the streams where we find them, summer temperatures will top out around about 24 degrees. And if it gets much hotter than that, they'll be extirpated. Okay, so yeah, there's plasticity, but will it help fish at the southern extremes of their species distribution? Probably not. But it may be quite useful for fish in the middle of their distribution where temperatures are starting to get hotter than what they've historically been used to, but they have the capacity to cope with it. But what about epigenetics? And so now I'm going to talk to you a bit about some work on stickleback. And this work, to be honest, was not actually conceived to think about climate change, but we started doing it because there was very little work in fish on epigenetics. So first I want to take a big step back and say, what is epigenetics? Because there's a huge amount of hype about it, but not so much knowledge. In fact, when we started on this work, it was one of those areas where you know there's a problem because the number of reviews was way bigger than the number of papers with actual data in them. So you know there's more hype than meat. So we thought, well, it's time to just sort of make some fundamental measurements and figure out what's going on. Okay, so what's epigenetics? Well, first, this is something, a little bit about the hype. So so this is a cover of Time magazine. It says why your DNA is not your destiny. And the idea is that it's certainly in humans and in medical research models, more and more they've been finding that the environment you experience during early development or the environment experienced by your parents can influence the phenotype of the offspring as adults. So these are long-lasting effects on phenotype. And this has huge implications, right? Because that tells you that, well, like it says on the Time magazine thing here, your DNA isn't necessarily your destiny. Your genotype is not the only thing that's specifying your phenotype. There are also these long-lasting effects that can affect phenotype. And then, because our society is what it is, there's been a ridiculous amount of hype around this. So, it boggles my mind as to what epigenetic yoga therapy might be. Epigenetic shampoo, skincare products, that are matched to your epigenome. It just doesn't make any sense. But nevertheless, there's this massive amount of hype, but it's actually quite controversial because we don't really understand very well the fundamentals of how epigenetic mechanisms work. Right now, we think there's sort of three major mechanisms that are involved in making these long-lasting modifications to phenotype. The one I'm going to talk about today is DNA methylation. So this is taking a methyl group and adding it onto a cysteine residue in the genome, and that changes the way that the transcriptional machinery reads the genome, so it changes the way genes are expressed. Similarly, you can modify histones, which are these proteins that DNA is wrapped around, and that changes, again, the accessibility of the genome to transcription factors, so changes gene expression. And then also microRNAs, which also regulate gene expression, can also act as epigenetic molecules, because all of these molecules can be passed along through cell divisions, and at least some of them can be passed from one generation to the next. So there's the potential for not just maternal effects, but also paternal effects that can be carried through. So when we think about epigenetics, we're living in kind of this time scale here. There are actually short-term plastic effects. I'm not going to talk about them today, but they're large. But there are also effects that can persist through development, and there are effects that can be inherited. And like I say, most of this work comes from medical models. So we were interested in asking, and there's also some really beautiful work in plants. So if any of you are plant ecologists, you may know that in Arabidopsis there's good evidence for epigenetic marks being passed through about more than 12 generations. So it's a thing, but certainly in fish we didn't know too much about it. So the first experiment I'm going to talk to you about is one where we wanted to ask, is DNA methylation, so one of the epigenetic marks, affected by early environmental temperature exposure? So in this case, we took stickleback, we took their eggs, and then you can, with stickleback, you can rear their eggs to hatch just in petri dishes. And so we took petri dishes with eggs in them, we took a single clutch and split them, and we did this for multiple families. And we raised them until hatch. And notice it says at different temperatures, 12, 18, and 24 degrees. And notice that it says 5 to 23 days. And that's because temperature affects developmental rate. So at the highest temperature, the fish develop really quickly. At the lowest temperature, the fish develop very slowly. But once they hatched, we put them into tanks and held them all at 18 degrees Celsius. So at a common temperature all the way to adulthood, which in stickleback is about 8 months post-hatch, 8 months, 9 months post-hatch. This is before they were sexually mature, but they were definitely like little fish, And then we measured DNA methylation, and we use a technique called reduced representation bisulfite sequencing, but it doesn't really matter. Essentially, we're sampling the genome for what sites are methylated and what sites are not. And then we can use bioinformatic tools to figure out what's changing. And you end up with a plot that looks something like this. So this is comparing fish reared at 12 degrees, or fish with reared until hatch at 12 degrees, with fish reared until hatch at 18 degrees. Remember, this is measured in fish that have spent the last 8 months at 18 degrees Celsius. Every dot represents a site in the genome that is differentially methylated, either higher methylated in the 12 degrees Celsius or lower methylated. And so basically there's lots of places in the genome, thousands and thousands of sites that change. And same thing if we compare 24 to 18. And the reason we always did the comparison to 18 is 18 is the temperature that stickleback are usually reared at in the lab. It's also the common, sort of the mean temperature in their habitats during their reproductive season. And then to ask whether this had an effect on phenotype, we used RNA-seq to look at gene expression. This was in the muscle of the adult fish. And indeed, we saw changes in gene expression. And so remember, these are fish that are experiencing, at that time, the exact same temperature. But back 8 months ago, for a few days, they experienced a different temperature, and now their phenotype is fundamentally different. So what we can say is that early developmental exposure clearly can cause persistent changes in both DNA methylation and gene expression. So that suggests that the environment you grow up in can influence what you're like as an adult. And work in the lab of one of my former students has also shown that it actually changes the amount of plasticity that you have. So how you develop can change how much of this short-term plasticity you express as an adult. Okay, but then the next question would be, all right, that's fine within the lifetime of an individual, but is there any evidence that that this kind of thing can be inherited? And sadly, we haven't done an experiment on this with temperature. Instead, we chose to look at the effects of maternal stress. And the reason why we picked maternal stress is that there's lots of evidence in a variety of different systems that stressing out a mother causes changes in the phenotype of the offspring. And in fact, it's now coming out more and more that you can also stress out the father and cause changes in the phenotype of the offspring. So it's not just a cytosolic factor that's being brought in the eggs, it's something that's being carried on the DNA. And again, we did this in stickleback. It was again a pretty straightforward experiment. We took females, we either left them unstressed, or we stressed them for 2 weeks, and we did what in the paper I think we called a simulated predation stressor, but in fact we chased them around with a blue guppy net. And and then we took eggs from both types of females, and we crossed them with sperm from unstressed males. So the same sperm was used to fertilize both types of females. We had multiple families generated. And then we reared the fish until, again, they were just prior to reproductive age, so they didn't have, stickleback don't have, only grow gonads in the reproductive season, so, or grow detectable gonads. So they didn't have active gonads or anything. They were just, little subadult fish. Then we took out their brains and we did RNA sequencing because we knew in other studies that there were differences in behavior. So for the difference in behavior, maybe there's differences in gene expression in the brain. And what we found was something that actually really surprised us, because almost all of the work that had been done in this field to this point on the behavior side of things only used male offspring, because a lot of behavior studies choose only to use males to, quote, reduce variation, because with females there can be, especially in mammals, there can be changes in behavior with estrous cycles. That of course is horribly sexist and just generally unwoke. So, so we decided, no, we're not going to do that. We're going to look at both males and females. And what we found was super surprising. And it's easiest to see probably in this Venn diagram on the left. So the numbers there are the number of genes that were differentially expressed. And so there are lots of genes that differ in expression between the brains of male and female stickleback. So those are the ones in the white circle that says sex. There were surprisingly few genes that were affected solely by maternal stress. That was a real surprise. We were expecting a big effect of maternal stress because that's what people had seen in other studies. But what was really interesting was that there was a huge number of genes that demonstrated an interaction between maternal stress and sex. And when I say sex, I mean the sex of the offspring. And you can see it here in this a heat map. So what we're looking at here is on the left are the expression patterns in the brains of fish whose mothers had not been stressed. And over here are the expression patterns in the brains of in the brains of the offspring of fish whose mothers had been stressed. And they're divided into male and female fish. And what you can see, so this is the interaction term genes. So you can see that fish that come from unstressed mothers Males and females have roughly similar gene expression. So if it's black, it means it's the same as the mean for that gene. But if we look in the brains of fish that came from stressed mothers, male brains and female brains are doing very different things. So if we look here, if we compare relative to the unstressed mothers, which sort of this group up here, the genes are mostly sort of in the mushy middle. The male brains are going down in expression of those genes, and the female brains are going up. So the same genes are doing the opposite things in male and female brains. And we can see this in this kind of plot here. This is a really weird plot. It's called a plot in semantic space. And what it's doing is, I don't know if you guys know what a GO analysis is, but it's a gene ontology enrichment analysis. It's asking what kind of categories of genes are represented in your data set. And you can plot these in semantic space, where genes that are more similar to each other in what they do are plotted close together. And each bubble is representing a group of genes with a particular name. The red bubbles are for females, the blue bubbles are for males. And so what you can see is that in males, what we're mostly seeing is increases in genes that do things like synapse assembly and nervous system development and various types of synaptic transmission and other signaling molecules. Whereas for females, it's mostly things that have to do with brain metabolism. So maternal stress is indeed having big effects on the brains of these sticklebacks, but it's having very different effects in males and females. And Alison Bell, who's a wonderful stickleback behavioral researcher, has, since we published this paper, gone back and looked and done behavioral experiments using the same kind of experimental design, but looking at both males and females. And indeed, she sees very different behavioral responses in males and females. So overall, what does this experiment tell us that might be relevant to how we think about how animals cope with environmental change? The first thing that we can see is that the environment experienced by the parent causes transgenerational effects. So what your parent experiences can change what you experience. So what we're doing right now is looking at these same things for parents that have been exposed to different temperatures. And this causes differences in gene expression and behavior. These effects differ between males and females. But the actual mechanism underlying this is unknown, because sadly, we didn't have enough sample from any of these fish to also measure DNA methylation in these same samples. We don't know if it's a methylation effect or histone effect or microRNA effect. But nevertheless, from the point of view of our sort of big picture question, we can say that there are detectable epigenetic effects in fish, both as a result of developmental exposure, so what you experience early in your life, and in terms of parental exposures. And so that potentially could allow organisms to rapidly respond to environmental changes. So then the next thing we want to think about is evolutionary adaptation. Can evolution rescue us from the extinctions that we're likely to see from climate change? And of course, the challenge that we have here in this adaptation thing is how quick is it going to be? Will it be rapid enough to cope with the rapid changes in the environment? Because really, environment is changing at an unprecedented rate. We know that the environment has changed over time. I'm from British Columbia. 15,000 years ago, it was under 3 kilometers of ice. It's still chilly, but it's not that chilly. So it's animals live there. So animals have distributions have shifted and so forth. But that took many thousands of years. We don't have many thousands of years for organisms to deal with anthropogenic climate change. And I know actually here in this department there are people who do beautiful work on rapid adaptation. And what I want to do is just sort of narrow in the question and ask whether traits relevant to responses to environmental stressors undergo rapid adaptation. And I'm going to give you an example using, again, 3-spine stickleback. And now I think it's worth thinking a little bit about the actual biology of the stickleback. So far, I've treated the fish as sort of black boxes, their ecology and so forth. I haven't even talked about, although all the work we do is deeply settled within their ecology. But why do we choose 3-spine stickleback for this work? So again, like I said, much of the current distribution of stickleback, so this is British Columbia right here, was under ice about 15,000 years ago. But today, you find stickleback in lakes and streams all over British Columbia. So those stickleback are thought to have originated from a marine ancestor that colonized freshwater. Stickleback have a very flexible life history. So some populations live out their entire lives, go through their entire life cycle in saltwater. There are other populations that undertake their entire lives in freshwater. There are other populations that breed in freshwater but then migrate to the ocean to grow to adult and then come back. And there's been a lot of beautiful work in this system on the morphological changes that happen. Because when you go from the marine environment to the freshwater environment, there's a ton of environmental differences that happen. So the temperature changes, the salinity obviously changes, and the extent and nature of predation changes. And we know that marine stickleback are heavily armored. They have armor plates, and they have large spines. Whereas in many freshwater populations, those plates have been lost, either to a small extent or to a large extent, and the spines are often much smaller. And the genes underlying those morphological changes are at least the major genes, are well known. This is beautiful work by Kingsley at Stanford and Katie Peichel and Dolph Schluter at UBC. But also, too, my lab was interested in asking, are any of these sort of climate change relevant traits being affected in these stickleback? And so some years ago we showed that marine and freshwater stickleback differ in their cold tolerance. So on the right here is actually their heat tolerance, their CT max. There's 2 marine populations and 2 freshwater populations. And you can see there's nothing much going on in the heat tolerance. But there's a big difference in cold tolerance. Marine populations are less tolerant, so they turn upside down at a higher temperature than do freshwater populations. And that's because in British Columbia, the ocean environment is a bit warmer and certainly more constant than is the freshwater environment, which gets very cold in the winter. We also showed that these differences in cold tolerance are heritable. So if you breed the fish in the lab across a couple of generations, the marine by marine crosses, and again this is from 2 different populations, still have not great cold tolerance, where fresh by fresh crosses have much better. And the hybrids, so this is a marine by fresh and a different marine by fresh, or this is fresh by marine, are somewhat intermediate, but actually tend more towards the phenotype of the ancestor of the marine population. So this exceptional cold tolerance is a recessive trait of some sort. Okay, so we know we have a system where in the past, after glacial retreat, there has been evolution in cold tolerance. So the question is, how quickly could that evolve? And so we did an experiment. And this actually, luckily enough, we were able to collaborate with Dolph Schluter, who wanted to ask the same question for the armor plating. And so what we did, and this is actually some heroic grad students, they went out to, this is a marine population, this is a marine environment here, this is a near-shore marine environment in British Columbia. And they collected stickleback from that marine population. And then they went through those fish and genotyped thousands and thousands and thousands of fish and found the rare individuals that were heterozygous for EDA, which is the gene of major effect that specifies the plate. And then we took those heterozygous individuals and used them as founder populations in a series of experimental ponds on the UBC campus. So they're manufactured ponds. They're ones that were dug out for research purposes, but they're natural ponds in that they have a natural invertebrate community. They're surrounded by vegetation. They're just ponds. They're just ponds we can control. So in went the fish, and we left them there. And every year we sampled them so we could follow genetic changes in the populations over time and phenotypic changes in the populations over time. And there's some really beautiful work on the morphological changes and on the genetic changes in these genes specifying the plates and so forth. And over the generations, we saw adaptation in exactly the direction we would predict for morphology. And interestingly, we saw the same thing for cold tolerance. So here's this heterozygous ancestor, which has sort of fairly sad cold tolerance as exactly what we would expect based on the data we had before. And here are the fish from the ponds after 3 generations. They are now statistically indistinguishable from fish that had evolved in fresh water since the last glaciation. So we can see here that within 3 generations, there has been, we presume, adaptation. It's possible that some of this is epigenetic, but certainly within 3 generations, we've driven the phenotype to a different point. So this suggests that rapid response is possible. Sadly, all of these samples were lost at some point in a freezer failure or something, and so we can't measure the DNA methylation in these guys, which makes me very sad, but these things happen. So, but we can conclude that certainly cold tolerance shifted within 3 generations of exposure to freshwater habitats that was accompanied by shifts in allele frequencies in genes known to be important for freshwater adaptation. So whether it's epigenetics or evolutionary adaptation, we know that it can happen rapidly. So that suggests that rapid evolution of thermal tolerance traits is possible, certainly for low temperature tolerance. This is not a great system for looking at high temperature tolerance because that is not different between the populations. But if you think back to how we did this experiment, we sort of cheated. We didn't start with just a regular marine population. We went through and we took out the rare heterozygotes for ETA. So in essence, we inflated the amount of standing genetic variation for the genes that are important. And indeed, we know that rapid evolution relies on the presence of standing genetic variation. Because if you are waiting for de novo mutation, you will be waiting longer than you would want, especially in a species like this that has 1 generation per year. So ideally, evolution rapid evolution works best in large populations with high levels of standing genetic variation for the relevant traits. So that really then brings us to the question of, is there evidence for standing genetic variation in traits involved in responses to environmental stressors? And so we'll go back to our Atlantic killifish that we began with, talking about plasticity. And remember that I just briefly mentioned that there were 2 different populations? That becomes important for this experiment. So this species is found in estuarine marshes along the Atlantic coast. It's found all the way from roughly Newfoundland in Canada down to northern Florida. And there's a very steep thermal cline along this coast. And then there's 2 subspecies that we know differ genetically, physiologically, and morphologically, a northern subspecies and a southern subspecies. That little graph on the left there just shows for a number of alleles, change in allele frequency with distance from the southern end of the species range. And so what you can see is that allele frequency more or less stays constant, and then we get a shift in gene frequency until we get to the southern form. So there's a sort of broad hybrid zone. Some alleles have extremely steep clines. This, you might be able to see, there's a red line right here. That's extremely steep. That's mitochondrial DNA. And then the others are various nuclear alleles that have clines of varying widths. Okay, why am I belaboring this? It's because these 2 subspecies have very different thermal physiology. The southern subspecies shown here in red has a higher CT max, so it's more warm tolerant, and also a higher CT min, so it doesn't last down to as cold a temperature as does the northern population. So and this is for several populations across the range shown in the figure here. So what we did is we went into the natural center of the hybrid zone. This is actually the center of the mitochondrial cline, so the individuals in this population are 50% northern mitochondrial genotype and 50% southern mitochondrial genotype. And we took those animals and we measured their CT max, and in this case we also measured their tolerance to low oxygen. And this just shows those data. So on the y-axis is CT max, and on the x-axis is their time to hypoxic loss of equilibrium. So basically we put them in a very low oxygen, we ask how long does it take them to turn upside down. And there's no particular correlation between the 2 traits, but there's lots of variation. So this suggests that there is substantial phenotypic variation in these traits. But the question is this genetically based variation, or is this some kind of an environmentally induced variation? So what we did was we used genotyping by sequencing to genotype these individuals, and we got about 20,000 SNPs. And by doing genotype-phenotype associations, we could show that about 50% of the observed phenotypic variation could be accounted for by genetic variation across about 30 of the SNPs. And we did this using a machine learning approach called random forest analysis. So that's pretty cool. So that suggests that there is substantial standing genetic variation in genes that are responsible for specifying these traits. We've done similar work in cultured Atlantic salmon that shows the same kind of thing. And in fact, the genes we turn up in these sorts of studies are things that make some sense. So this is one of our sort of favorite genes that we're following up from the genome-wide association study. Something called an E3 ubiquitin ligase, and it's a gene in the pathway that's responsible for clearing out proteins that are damaged by high temperatures. Okay, so that suggests, at least in killifish, there's substantial standing genetic variation in thermal tolerance traits. But maybe killifish are unusual. We were in a hybrid zone between two subspecies that differed in their thermal tolerance traits. Maybe, again, like in the case of the stickleback, we were sort of stacking the deck to find high levels of this. So this leads me to a project that we're currently doing right now. It's a big team project called Sustaining Freshwater Recreational Fishing in a Changing Environment. And the whole goal of this is to provide information for the sustainable management and conservation of rainbow trout. And so we're doing things like looking at genetic variation across the range and associating it with landscape features, so environmental temperature and so forth. We're looking at connecting genotype and phenotype. We're developing specific genomic tools that management can use. And we're also working with social scientists to actually make sure that managers actually do use the tools. Because one of the, I'm sure for those of you who work in conservation science, it's all very well to derive science-based information, but if no one actually uses it, then it's been a waste of everybody's time. So why rainbow trout? Well, for one thing, rainbow trout are economically, socially, and culturally important in British Columbia. So they're commercially fished. This represents an example of First Nations or Aboriginal fishing. There's a very large recreational fishing industry. They're also very important to First Nations culture. They're also threatened by environmental change. So these are just, this is a sign on a river in British Columbia that it was closed to fishing because of high water temperatures. There've also been like multiple headlines where they've just had to do fisheries closures because the water was getting too warm. The fish could survive, but what they couldn't survive is the stress of catch and release angling. So, because most of this fishing in the rivers is catch and release. So the idea with catch and release is ideally when you put the fish back, it's supposed to survive. Sadly, a lot of the time they don't, and certainly when the temperature's high, they don't. So rainbow trout, they're found on this coast. They've been introduced throughout the world, so we're also working on some of the introduced populations. We've been using whole genome sequencing to identify the levels of variation, and there is a lot of variation among these populations. Even if you take, say, two lineages from the northern interior, you will find 20 to 30,000 SNPs between different populations. And so you can see these clusters in these big sort of PCA type analysis. However, there is limited variation among the strains in their thermal tolerance. There is some, it's detectable, but it's not huge. But there's very substantial variation within strains. So these are 3 British Columbia strains. And on the y-axis is the CT max, the highest tolerated temperature. Again, on the x-axis is the lowest tolerated oxygen saturation. And so you can see, this is a domesticated strain. The 2 traits are not very correlated in the domesticated strain. The 2 wild strain, these 2 traits are really correlated. So you have some individuals that can tolerate quite high temperatures and quite low oxygen, where other individuals that don't tolerate very high temperatures and don't tolerate very low oxygens. And so right now we're in the process of genotyping these animals. We've developed a 200,000 SNP chip, and with any luck we'll be able to soon tell you something about the genes that are involved in this variation. Okay, so I think now for both killifish and trout, we've shown you that there's a certain amount of variation, and at least some of this is genetically based. So overall, kind of what I've shown you today is that there is definitely plasticity in existing populations. There's a potential for epigenetic effects, and there's a certain amount of standing genetic variation that may allow some evolutionary adaptation on timescales that may be relevant. On the other hand, we don't know whether any of this is going to be enough, especially at the southern limits of a species range. It may allow species to persist in areas that are currently in the middle of the species range that will eventually become the southern limits. But certainly I think, or at least I hope, that I've demonstrated to you the importance of thinking about these questions across multiple timescales with multiple mechanisms in mind. And with that, I just want to wrap it up, thanking the people who actually do the work. At this point, I'm mostly the spokesperson. Every so often I get to go out in the field. This is one of our field sites. And help out, but it's these people who do the work, and of course, the agencies that provide the money. And I thank you very much for your attention. And if anyone needs to go, feel free, but I don't know if we have a minute or two for questions. Yeah?
How do you, I'm not clear on how these results translate into a genetic management plant? Do you maximize genetic diversity in population? Is that your criteria, or do you actually look for a specific set of genes and mutations that seem to do better across the
Right now, that's actually one of the things that we're working on with the social scientists who are interviewing managers, because right now the genetic management plan is very much, because rainbow trout are stocked really extensively, so they're very actively managed, and the approach is very much, to the extent that they can, stock with local genotypes. But the challenge is that works fine so long as the local environment is not changing. And then so then the question is, and that also only works if, or works best if you're maximizing the genetic diversity, the local genetic diversity that you're stocking. A lot of the reasons we've gotten into trouble in the past is that there's been unintentional hatchery selection as part of the stocking program, and so that what they're actually stocking is not well suited for the environment. Going forward, it may become necessary to start stocking with non-local genotypes that have, or the rare individuals in local populations that have the most desirable cassettes. This is extremely controversial in the fish world. It is less so in forestry, because you think about, if we have problems with this in terms of thinking about adaptive management in fisheries, think about it in forestry, where if you're replanting an area or if you're managing a forest, you're planting things that are going to have to survive for 100 years. So you have to think about not only current climate, you have to think about climate 100 years from now. And so there's very active discussion in adaptive management in forestry as to, it's sometimes called, what is it called? Assisted migration. So the idea being that instead of planting the local genotype, you plant a genotype from 100 kilometers south or 200 kilometers south. And that is essentially the equivalent of what would happen if the organism were able to migrate to that spot. And with trees, that may be possible, because their dispersal is really is across great distances. With trout, it's potentially a problem, because a lot of these are lake endemic populations, and so they are unlikely to be able to move rapidly. Steelhead, on the other hand, there's some potential for movement of genotypes around latitudinally. So that's not a great answer to your question, but it's certainly something that the field as a whole is grappling with. Anyone else? Yeah.
You mentioned hatchery selection. I wonder
Yeah.
if is there thinking about using temperatures in hatcheries to pre-adapt during the developmental stage to the same genotypes into the right environment?
Yeah, absolutely. So they're looking at that. Often it's actually often at the moment the problem is actually that in hatcheries they often keep the temperatures kind of high to accelerate growth rates to get the fish out sooner, and sometimes that can actually cause problems. So one of the things that we're doing actively in the lab with a variety of species is working with hatcheries to try and optimize production approaches to improve things. I was telling a couple of the grad students I was working with that we're doing some work with sturgeon, and they're really a problem, because number one, they're highly endangered. Number two, depending on the species, it may be 10 years before they first reproduce, and number three, as far as we know, almost none of the hatchery-reared sturgeon are recruiting into populations. So whatever they're doing in the hatchery is not working. So yeah, so it's a huge problem. There's also really good evidence in some of the salmonids, not work done in my lab but in other labs, that there are major epigenetic effects of hatchery rearing, and that may be causing some of the problems. And so figuring out what needs to be done to deal with that is a huge problem. Yeah, so I've been working on these general questions for a long time. A good probably 15 years ago now, we worked on Atlantic salmon in the Bay of Fundy, which is on the east coast of Canada, and there they have these massive hatchery programs. They're releasing hundreds of thousands of fish, and they get returns of like 2 fish. So it's a huge amount of government money going into trying to maintain these populations, and it is just not working. And now part of that is hatchery based. Part of it is changes in the marine food webs that are just making it difficult for these fish to recruit. But the wild fish, the few remaining, do seem to be recruiting better. So yeah. Yep.
your experiments that were looking at temperature of CT maxes,
Yeah.
you mentioned that 35 degrees, they can limit that to not for
The Killies, yeah.
period of time. How do you take that time component into your analysis? Because I can see CT maxes being an acute for a
Yeah.
chronic over a period of months.
Yeah, absolutely. So today I mostly showed you data on CT max, which really, if you think about it, that's a trait that will be important for those brief high temperature excursions. But in fact, the other thing that we're working on is fundamentally what you really want to know is like what's the maximum temperature at which there's effective reproduction and growth rates. They're just harder things to measure in the lab because they take longer to do, so we don't have as much data on them, but there's no question that that's where it needs to go. Because, I mean, I sort of basically implied this just a little bit with the data where we're looking at the CT max and the oxygen. It's not one trait that you want to select for, right? Because if you just select for CT max, God only knows what trade-offs there may be. And maybe these individuals that have very high CT max have very low reproductive output or something else. So right now, we know so little that it's very difficult to figure out how to do these things. And so we're just kind of scratching at the surface of the big picture problem. But you're 100% right that this is the essence. It's trying to figure out what are the series of traits, what are the series of genes that we should be looking for in managed populations. This is why life would be much easier if we had a world that was not changing, because then we could let evolution do its thing, because most of the time when we try to mess with it, we don't do a great job. So there's another question up here, I think in a sec. Yeah.
Hi.
Hey.
I'm curious if you've thought about like shifts in thermal tolerance in terms of invasibility.
Yeah.
Yeah. One of the big things is like introducing different fish in different areas where they're not endemic.
Yeah.
Due to like game fishing or just the aquarium trade.
Yeah. Yeah, it's a really interesting question. We haven't addressed it directly, but it is worth looking at. Again, it's just one trait, and there's many others that will influence invasibility, but there is some evidence in other species, again, depending on the environment, that animals with high resilience tend to establish pretty well. And that's sort of what you would expect because they're able to cope with a range of climates or habitats. I think there was another one there. Yeah.
I was curious about what you said with there being limited variation between strains that we're
Yeah.
talking about. And if you had any thoughts as to why that is, because
Yeah, because there's huge genetic, yeah.
rapid evolution.
I mean, I actually think that CT max may be quite constrained and that it is unlikely to undergo rapid evolution, because I think a lot of populations have been pushed kind of as far as they're going to be pushed. I mean, there's clearly some variation, so and some of it is genetically based, but it's not huge. Like if you look at it, it's a few degrees Celsius, right? And if we go back to the strain variation, that's maybe on average, half a degree Celsius. So I think other traits are more likely. Again, we started with this one because it's easy to assess, and the annoying thing about these genotype-phenotype association studies is you need thousands of individuals. So you need a trait that you can measure on many, many individuals. So it's a place to start, but yeah, I think for rainbow trout, that's not going to, finding populations with extreme high CT max is probably not going to get us out of the problem. I remember I mentioned that they've been introduced around the world. We've been working with Western Australia because they've introduced rainbow trout there, and it's really hot in Western Australia. And so they have a series of populations that they've been doing in their hatcheries that have undergone essentially unintentional selection in the hatchery for high temperature tolerance. And their CT max has shifted a little bit, but there's actually been quite large shifts in their cardiac performance with temperature and their growth performance and that kind of thing. So we're working on those fish to try and figure out what's different about them and what's the genetic basis of those differences. Yeah. Any last questions, or should we call it a day? Well, thank you very much for your attention.